And today you're going to be learning about how scientists use information that they get out of the earth, or either from looking at cliffs on the side of the earth, to help them understand how organisms have possibly evolved over time, or when they lived and when they disappeared. So, talking about drill cores, stratigraphic columns, and a few other things. So drill cores are actual samples of ice or soil that have been taken in vertical columns out of the earth. So literally what scientists do is they use a big drill bit that will take a column and go straight in the earth. And they can actually go miles deep with these things and pull them up one layer after the other after the other. And what they pull out can look like this. This would be one that was taken out of a hard rock. And so that's why it's so nice and pretty and neat, and you can see every line perfectly, but it gives you a really good idea of what scientists are looking at as you look at the different layers. So as you see them, you can see all the different lines. Every layer, every pretty little line that you see in a rock like this when it's taken straight out of the earth is rock that was laid down at a different time period in the Earth's history. And so as scientists look through these rock layers, or through these dirt layers, or even they can do it in ice. Ice has a lot of history in it. But as they look through these layers and they look at the organisms that were found in each layer, they can determine when organisms lived, how long ago they lived, and even when they disappeared, when the organisms quit showing up in the rock or in the layers of the dirt or ice. Now, most of them, again, they look more like this. It's dirt, kind of crumbly, so scientists have to be careful with them. But ice can also be used, and actually a lot of the information that we have about the climate history of our planet comes from ice cores. In areas like Greenland, the ice can be over 2 miles thick, and to get 2 miles of ice takes thousands upon thousands, even millions of years. And so as scientists study the gases that are trapped in the bubbles that are found in the ice, the scientists can get a lot of history of the earth. But that's what a drill core is. And once scientists pull these drill cores out, they can use them to make stratigraphic columns. All right, and what a stratigraphic column is a diagram that represents that series of rock layers. And they can make it by looking at the drill cores, so looking at those layers, or by looking at a cliff face. If you've ever seen pictures from out west, the painted desert with all those beautiful layers through it, every single layer represents a different time in the Earth's history. And so scientists can look at the sides of a cliff, might look something like this, and see when that cliff was laid down in history. And then they can use information from radioactive dating and things like that to actually see when those periods were laid down. And now they'll have this nice drawing, this sketch to show the history of the earth in that area. And so that's what a stratigraphic column is. Now those stratigraphic columns can be used to figure out what the oldest organisms are and what the youngest organisms are, because of something called the law of superposition. And what the law of superposition says is basically, if you're looking at a sequence of rock layers, now again, that's whether it's a drill core or whether you're looking at the side of a cliff. If you're looking at a series of rock layers, okay, whatever is on the top, so up here, along with anything found in that layer, any fossil that's there, is the youngest era. Okay, that's the youngest time period. Those are younger than anything else. And the deeper you go, the older the organisms get. So the youngest is at the top, and when you have a drill core or a stratigraphic column, whatever you see at the bottom is going to be the oldest. And that is because as layers of earth are made, the new stuff is laid down on old stuff. So what's down here gets covered up by this layer, and then this layer gets covered up by this layer, and then this layer gets covered up by this layer. And so the oldest is always going to be at the bottom, while the youngest is always going to be at the top. And you can use that information to organize things, living things, plants or animals, based on which one's the oldest, what's found down here at the bottom, and which one's the youngest, what's found down here at the top. Now, if you'll bear with me moving you for a minute. Give you a little bit of a demonstration of how you can use these cores to correlate one sample to another. So one of the things that you're going to be doing is an activity where you have drawings of organisms in rock layers, something like this, that are taken from supposedly different areas of the planet. And your job is to match up one layer to the next to figure out how they would match side by side. Because see, when you have stratigraphic columns from different places on Earth, the dirt might look different, the soil might be different soil types, but if you have the same organisms, okay, living there, you're looking at something from the same time period. Because that might be a different dirt laid down over there, and it might be deeper over there, or it might be shallower, but if the same organisms are there, you have the same time period. And that's what scientists do to figure out how old or how young or when something was. They look for those index fossils, those special fossils that line up. This is my bad drawing of a trilobite. This is an ammonite. Okay, it looks like a snail. This is a fern, and this is a fish. And so what you will then do is you will try to match up matching rock layers, the ones with pink, and matching organisms. So like here we have pink and an ammonite and a trilobite, and here we have pink and an ammonite and a trilobite, and this doesn't quite match. See, it's white. But if I slide this one down, I have pink with an ammonite and a trilobite. Okay, and I have a white and a white and a white, and ammonite, ammonite, trilobite, trilobite, I'm still okay. Then I have a pink and a pink and a pink, and they're all got ammonites, and then I got a white with ammonites, and I got a fern here. And so maybe this one's just a little bit off. Maybe I need to go up just a little bit. And now I have my pinks and my whites and my pinks and my whites, and they're still kind of matching up. Now I have an ammonite here and none here, but that's okay. You might not have everything in every layer. The key though is I have fern and fern and fern. So I'm okay. Even though I have a fish over here and not in these two, and even though I have an ammonite here and not these, it's okay, because then I'm back at white, 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 and I've got a fish, and a fish. So I'm still okay, and now I've slid this one down, and I've slid this one up a little bit, and now they are correlated. And that is what you're going to be doing in your activity when you try to match up the drill cores that I have drawn for you and that you see on your paper. Please let me know if you have any questions. Send me an email, send me a snap of what you're having a question about. I'll try to help you answer them. Thanks.